In contrast to aerobic organisms, strictly anaerobic microorganisms require the absence of oxygen. As oxygen is ubiquitous in air, retaining oxygen-free conditions during cultivation is a prerequisite for anaerobic culturing.
The applied technique represents a simple and inexpensive method to provide anoxic conditions to cultivate a mixed microbial population in liquid culture.
This technique can also be applied for the cultivation of pure cultures. However, for highly oxygen-sensitive microorganisms, this procedure might not be applicable.
Start by weighing medium ingredients in an appropriate flask and dissolving them with half volume of distilled water. Then add 1 milliliter of redox indicator followed by vitamins in trace element solution. Adjust the pH according to organism requirements and bring the final volume to 1 liter with distilled water. The color of redox indicator is pH dependent and might require some time to adjust. Fill 120 milliliter serum flasks with 50 milliliters of medium then incubate them in a 100 degrees Celsius water bath for 20 to 30 minutes to reduce the solubility of oxygen in the liquid phase. Flush the headspace with nitrogen.
Take care of appropriate room ventilation when working with nitrogen.
Close the flasks with butyl rubber septa and fix the aluminum caps.